Okay, we have a couple of minutes, so while people are getting into the
Okay.
class, I want to share with you some cool things that I've been learning
The
with
new
using AI
one
to help
tomorrow.
me study. Can you guys hear me there?
Yes, everybody can hear you.
Oh, all right, cool. Okay, so before we go into the PowerPoint for today, I wanted to show you, let me see here where it is. That is artificial intelligence, right? At FAU. I assume everybody's familiar with this resource, hopefully. Okay, so what happens here? You go here, or say students or staff, so you go here to this AI resource and you can check out some cool AI tools. And here, you're gonna find several tools that I don't even know how to use yet. I know that several professors use this Copilot. I don't know how to do that yet. But I learned about this other one all the way down here. This guy here, Notebook LM. Who is familiar with this tool? Let me see, like show of hands in the class. Did I see hands going up?
We've got like one. If you asked who's familiar with ChatGPT, I think the number would be different.
All right, ChatGPT
Tuna,
is
by the
also
way.
good, hopefully. Who has used
Mhm.
ChatGPT? All right,
Yay!
he knows
All the
about
hands.
ChatGPT. All right, so you guys gonna like this one here. Okay, check this out. So you go to Access Notebook LM, you click that, and then you say try Notebook LM. Okay, so my page is gonna open here,
Okay.
and I have a couple of notebooks that are open there. So let's say that if I want to create a new notebook, this is a bit tricky, okay? Because first thing you have to do, you have to upload a file. So you have to have a file in PDF, okay, that you need to upload. I'm not gonna do that. So I'm gonna show you here an example or what I did with my other class, okay? So here I have my cell biology class, and yeah, the number of slides is almost as many as we have for this class. So here I have 73, right? So you cannot just upload a PowerPoint or a PDF that is 73 slides long because that AI is not going to process all this information in a very good manner. So what I did here is that I selected a topic. Okay, so I selected here, for example, model organisms. So I took 11 slides that described everything about model organisms from this class. Okay, and I made a PDF and I uploaded the PDF like right there. Okay, so I'm gonna go here and show you what happened. So once I uploaded the PDF, which is, average 10 slides long, first thing this will do, it will give me a summary of what is in that PDF. And then you're going to have choices of these four things over here. So I did not click on reports yet, but basically you can make a study guide out of this. Look at this. If I click on generate a study guide, so it will come up with a study guide for this topic. Okay. Now what I clicked before was this mind map. Check this out. So I go here, model organism mind map. Okay. So first is going to tell me what is the definition of the model organism. So I can click on that and this will show up. Okay. So model organism is a whole thing, a whole living thing that can be used for studying cellular processes. Then you can close this and you can open a second subtopic of this slides. It'll give me the organization. I mean, isn't that cool? So unicellular eukaryote, you give me the model organism for that. You'll give me the studies on bacteria and viruses. So this is the mind map, okay? Now, one thing that I really like about this tool is that you can make podcasts out of this. And at first I thought this was going to be something really boring on a monotone kind of, reading the book out loud is going to put me to sleep. But no, this is really cool stuff. Look at that. If I go to the podcast, listen how this
Imagine trying to
comes out.
understand a massive, intricate machine by studying every single screw at once. Just overwhelming, right? Completely. But what if you could pick out a smaller, simpler version of that machine, learn its fundamental workings, and then apply those lessons to the big one? Well, that's pretty much exactly what scientists do to unlock the secrets of life. It's a really powerful strategy. Today, we're taking a deep dive into the world of model organisms.
I mean, isn't that neat? It will give you this podcast that is basically a talk between these two very human sounding creatures. And it will explain in simple terms everything that I have included in that PowerPoint. All right, so this is a very cool tool that I think you guys are going to like it a lot, right? Besides, you can also have new videos made up from those slides, and you can also have study guides that you can use to help you, better understand the class and do better in the class. All right, so those are my 5 minutes on what I have found out about AI. So now let's go here to our PowerPoint for the class. And we had a few slides that were left from last class, but those slides that are just a review of basic information that we have for the last lecture, I have included that in the pre-class video for nucleic acids for this class. Okay, so I'm just going to zoom very quickly through what you need to know about these slides, but make sure you watch the pre-class video. So just as a show of hands, how many people have seen the pre-class video? You really gotta, raise your hand like kind of high because, okay, I see some arms going up. Okay, everybody else who did not watch the video, make sure you watch it. It's going into a lot more detail than what I'm gonna fly through over here. Okay, so these last few slides on nucleic acids, basically we should know that they have nitrogenous bases and sugars and phosphate molecules. Okay. And the nucleic acids, they are going to form up monomers. They're called nucleotides. And yes, you have to know the difference between nucleotide and a nucleoside. Okay. And you have to also know that the nucleotides that make DNA is the deoxyribonucleotide type. And the ones that make RNA is ribonucleotide. Okay. So you should know the purines, what they are. So those are the same for DNA and for RNA. So that's adenine and guanine. No, you do not have to know the structures, but you have to know generally how big they are in comparison to the pyrimidines. And I have a little mnemonic for that. Okay. Now, pyrimidines that go into DNA, they are cytosine and thymine. So here's your basic structure for a nucleotide. And nucleotides will hydrogen bond with each other. So remember what the hydrogen bond is, right? Is this non-covalent type of interaction or attraction between a electrophilic element and hydrogen, okay? So here is your hydrogen bond. Now you're going to see that depending on the nucleotide, you're going to have either 2 or 3 hydrogen bonds. And now if you pay attention to this molecule, you'll see that you also have a hydrogen over here, and you have oxygen over there. And then the question here would be, why is it that this hydrogen is not forming hydrogen bond to that oxygen? You see? I mean, here you have hydrogen, oxygen. Here you have hydrogen and nitrogen, which is also electronegative. But then here, You don't have a hydrogen bond between this oxygen and that hydrogen. Does anybody know why? No? I'm
We might
assuming.
have,
Huh? Do we have a guess?
yeah, I'm trying to listen to it. Hydrogen can only have one bond, and the object, the octet of the carbon. Are we on the right track, or
No.
there's no point in finishing the sentence?
Hydrogen will make one covalent bond. But then look at this. Covalent bond on this side, hydrogen bond on that side. Huh. Covalent bond here, hydrogen bond there. Hydrogen bond is not a covalent bond. It's just interaction. Okay, it's attraction. Now look at this. This guy here is bound, covalently bound to carbon. Carbon is not very much electronegative. Nitrogen is electronegative. Nitrogen, and oxygen, nitrogen, yeah. But carbon is not. So this guy here is not going to form a covalent bond with the other oxygen over there because it is covalently bound to not very electronegative carbon. Okay, so you were right mentioning something about carbon, but what you're missing was the electronegativity. It's not strong enough to make this hydrogen to make a covalent bond. Because if this carbon was very electronegative, like this nitrogen over here, it is going to try to steal, to take away the electron from hydrogen. Okay, so it's electronegative. Now, if hydrogen is having its electron being taken away, it is going to want to get together with the electron that is popping out, that is showing out here from the oxygen. Okay, so this is how you get that hydrogen bond over there. So these are details on what's happening with the chemical composition and the chemical interaction that sometimes we just look in the book and you say, oh, A binds to T, two hydrogen bonds. C binds to G, three hydrogen bonds. But you don't really understand why. Okay, so this is why. Because in order to make a hydrogen bond, this hydrogen has to be covalently bound to electronegative atom. Okay? All right, so a little spiel on those hydrogen bonds. And what is going to happen here, you have the hydrogen bond at the center of your double helix. You have the backbone of your double helix that is made up of your sugar molecules, right? You have phosphodiester bonds that are linking those together. We're going to see that into more detail. Okay. And again, A binds to T and G binds to C. Okay. Then when you look at the RNA, ribonucleic acid, we see that the base sugar is not deoxyribose, but it is ribose. And you don't have thymine, you have uracil. And here comes your first iClicker question. Let's see if everybody is awake and ready to get those points. Can you start the iClicker, please, Ray? The question is, the major building blocks of nucleic acids are... Oh, I should make this more difficult.
Do you want me
Alright.
to like start this iClicker or just start the program? Because I did it wrong last time. So
Do the one that is the right option. I don't even know.
like, do you want me to start the poll
Yeah,
so they can answer?
yes. Start the poll because I have no idea what's going on over there.
All right. Okay. 30 seconds, everyone.
Okay, so I'm expecting everybody to get the 2 points. Actually, the first question is always 3 points because you get 1 point for participation, for being in the class, another point for participation, and a third point if you get it right. So everybody should come up with 3 points out of this one.
5 seconds. All right.
Okay, what's your answer?
C.
Yes, here is nucleotides. All right, so here, cool slide on structure of nucleotides. Look at this guy here. See this basic structure here? This little guy with only one ring, that's your pyrimidine. It kind of has a Napoleon complex. So because it's small, it wants to have a big name, kind of what I mean. Like the guy is short, wants a big name to make it look bigger, Little guy, big name. Bigger guy, smaller name. So this guy with two rings, that is your purine structure. So purines, you have adenine and guanine. Now, you do not have to know exactly the molecular structure of that, but know that there are two rings. And then pyrimidines, you have cytosine, thymine, and uracil. So what is going to make your nucleotide? You're going to have your sugar. You're going to have your base. So this is your nitrogenous base, which is going to be one of those, right? And then you're going to have a phosphate. And how you're going to remember the structure of your nucleotide? Well, your phosphate is your phosphate that comes from a phosphate pool. So let's think that this is a pool, like you go and you swim in a pool. So the phosphate pool, and the base, the nitrogenous base, that's going to be your garage. Okay, so the base for your car here is your nitrogenous base. And then the house, the little house shape, that is your sugar component of your nucleotide. So in order to be a nucleotide, you need to have at least one phosphate. Okay, if you do not have a phosphate, then this thing here with sugar and base, that is called a nucleoside. Okay, with the S here, change the T for the S. All right, so nucleotide, at least one phosphate. So if you put two phosphates here, that's still a nucleotide. If you put three phosphates, that's also a nucleotide. Okay. So how are you going to remember which ones are your purines and which ones are your pyrimidines? I like to put this guy here, the Pikachu, maybe Pikachu, but if I use Pikachu, then it doesn't make sense. So this guy here in my slide is called Pugachu. Okay, so purines are guanine and adenine, and they have two rings. Okay, so now you just have to remember the other ones have to be pyrimidines. So now looking more details on these nucleic acids, we know that these nucleotides are going to give you a lot of energy because they hold energy on their phosphodiester bonds here. Okay, so these bonds here are both not stable. They are they are quite unstable covalent bonds. Okay, so how do we know that they are unstable? Let's look at this. This phosphate group right here, it has a phosphorus in the middle and it has oxygens all around. And the oxygens are very electronegative and they carry electron clouds all around them. So the whole molecule here, this whole piece of the molecule, it is very much negative. Now you stick it to another very much negative molecule. So negative electricity, negative charges here, they are repelling each other, but still they are connected with their bond. And then you put another negative bulky thing on the other side. So they're going to repel each other even more, and they still have together by these covalent bonds. So what's going to happen here? If you manage to break away this covalent bond, actually, I think this one should be over here. Well, first, how are you going to break away that bond? You're going to have to use an enzyme. You're going to have to use a form of reaction that is going to add a water molecule. So this is going to be a hydration reaction, hydrolysis, breaking lysis. So breaking with water, this is going to be a hydrolysis reaction. Yeah, that sounds like a test question. Okay, so you do a hydrolysis reaction right there. Then you break that bond, but by breaking the bond, you're going to liberate all the energy that that bond is holding. So this is why ATP is the energy currency of the cell, because you have enzymes to break the bond and liberate the energy that is in the bond. Okay? All right. So here is your reaction. When you hydrate or do hydrolysis of ATP, you break away one phosphate, you end up with ADP, so diphosphate here, and energy that was held in that bond. Now, when you put the phosphate back into ADP to make ATP, now that is going to happen mainly at the inner membrane in the mitochondria, right? And that is going to store energy in the ATP. And the energy used to store that energy, that's going to be your electrochemical gradient, if you remember your ATP function from another class. Okay? All right. So examples of work that are driven by this energy that is found in ATP. Okay. So you can have transport of things through membranes, and not only transport, but this thing is going against its concentration gradient. So you need energy to push something against the concentration gradient. Another example is mechanical work. So if you're going to contract muscle cells, you're going to have to use energy for that. And another example is also synthesis or anabolic reactions. You do need to use energy to come up with bigger molecules in anabolic reactions. Okay. So now we finally get to chapter 3, and this is going to do, has to do everything about living cells, right? Cells are the smallest living units. Okay, so breakdown of that chapter, we're going to start looking at the plasma membrane. And after that, we're going to see how useful is the plasma membrane. And we're going to look at transport through the plasma membrane. And you're going to have to know two types of transports. One of them is active, and the other one is passive. Okay. Besides that, you have to also know how the membrane gets to voltage potential and how the inside of the cell gets to be more negative than the outside of the cell. So after that, we're going to look at the cytoplasm. We're going to see the whole mush of stuff that is inside of the cytosol. And you're going to look at organelles that are in the cytosol. And then we're going to look at the nucleus and briefly the structures in the nucleus, and then how important it is for the nucleus to be separated from the cytoplasm. Okay? So we know that cells are the smallest living things. So if you go into anything smaller than the cell, then the stuff is not living. And yeah, I had to have hiccups. Yay. All right. So if you look at viruses, they are not living because viruses depend on a living thing, on a cell, for the replication. Okay, so the virus can invade the cell and use the cell machinery to replicate, but on its own, a virus cannot replicate. A virion cannot replicate. All right, so the cell is the smallest living unit. So we're going to see about the structures and the functions of the organelles that are in the cell, and how their structure really complements the functions for these tiny organelles that are inside of the cells. Okay. So we look at the different types of cells that we have in our bodies. We know that every one of those cells originated from one fertilized egg, right? So we have one cell with the proper set of chromosomes, and that cell divides, and from that initial cell, you get differentiation into all types of cells. So again, when you look at a cell that is in your skin and you compare that to a cell that is in your liver, you're going to see that the nucleus has the same number of DNA because those are somatic cells, but the expression of the DNA, the expression of the genes is different. So the genes that you express in your skin cell is totally different than the genes that get expressed in the liver cell. And cells can only come from pre-existing cells. So if you don't have a cell to begin with, you cannot make a new cell. How many different types of cells you have in the body? Well, approximately 250 different types of cells. Okay, how many cells you have in the body? Well, a lot. Okay. So diversity of cells. What kind of cells you have in the body? So here are a few examples. You have cells that are going to help with composition of fibers of connective tissue. So you have these fibroblasts. You have certain cells that are important for exchange or filtration of certain molecules. Or in this case, you can also find cells that are part of your skin. So those are cuboidal epithelial cells. You have transporting cells. So those are your red blood cells or erythrocytes. You have very long cells. So those are skeletal muscle cells. They're also called fibers. We also have other types of muscle cells. There are smooth muscle cells. So these guys here are voluntary. These guys here are involuntary, that you cannot control their movement. And you have defense cells. So here is a macrophage that is an example of your immune system defense cells. Then you have some storage cells. I don't like those that much, but they're necessary. So here's an example of a fat cell. And look at this. It's going to store a huge droplet or vacuole of stuff, mainly triglycerides or solid fat, right? So look at this. This is the vacuole storing. The nucleus is down over there. It's like squished on the side, okay? This cell basically doesn't even have a cytoplasm. All it has is like this huge vacuole, okay? Vacuole is when you have something that is enclosed in a membrane in a cell and is very big for storage. When you have little membrane enclosed compartments and they're very tiny, you call those vesicles, okay? Vesicles and vacuole. Okay, so there's a difference between those different storage things. Okay. And here is a very typical neuron. So this is a neuron, okay, for the nervous system. And this guy here is not a somatic cell. This is a germ cell. So here is your sperm. It's the only cell in the human body that has a flagellum. So here, looking deeper in what the cells have. So there are three main things that the cells have to have. Okay, one thing is the plasma membrane. So if you don't have the plasma membrane, or if the plasma membrane gets broken, the cell dies. Another thing is the cytoplasm, and that's the soup that's going to have all this stuff in it. Okay, and then you have, of course, you have to have your nucleus, right? And you notice here that the double membrane of the nucleus is continuous with the membrane of the rough endoplasmic reticulum. And here comes another question. The three main components, oh, I just gave you the answer. Three main components of all cells include the plasma membrane, the nucleus, and the, you don't need more than 10 seconds for that. You just start.
Right.
I
I'm going to
click.
start it.
All right.
20 seconds. Ten seconds. Get something in. Even if it's wrong, you get half credit for just putting something in. All right, what's the answer?
What's the answer?
B.
Cytoplasm. So that was inside of the cell. Now, when you look outside of the cell, you're going to have stuff that is outside of the cell. So you call that extracellular stuff. Okay, so what do you find outside of the cell? Well, if the cell is alive, it has to be bathed by fluids, right? So you look, for example, if you look at cells on your skin, the very surface of the skin is made up of dead cells. Well, how we know that? Well, basically, they're in contact with the air and is not liquid stuff, is not fluid. So if their cells are in contact with the air, they're dead. In comparison, when you compare with cells from your mucosa, like from the inside of your mouth, those are wet, right? Because you have saliva inside of your mouth. So those cells are alive. So cells inside your mouth, they are alive. They're bathed by fluids, okay? So extracellular fluids, what's in there? You have blood plasma, you have cerebrospinal fluid, and you also have interstitial fluid. That's the name we give to the stuff that is between the cells in your body, okay? The stuff that bathes your cells in the body. So interstitial fluid and the blood plasma, okay? Blood plasma is when you take whole blood and you spin it down, the cells go down from that suspension, the top clear part, that's your blood plasma. And another example of extracellular fluid is cerebrospinal fluid that is bathing your brain and your spinal cord. It's surrounding your central nervous system. Okay. What else you find outside of the cells? You're going to find secretions. So saliva and mucus, those are secretions. And you're also going to find extracellular matrix, okay? So the stuff that holds cells together, or the glue, gooey stuff that holds cells together. So this matrix tends to be jelly-like. And if you start to think about that exclusion zone, okay, the easy water, that would make sense because the matrix itself has charges. Okay, so that would form easy water and that gel-like state of water. Okay. So we know that about 60% of the body is fluid. So that's a lot of fluid if you compare in a 70 kilograms person. 70 kilograms is about 140 pounds. So you have about 84 pounds of fluid in a 140-pound person. Okay. So again, what are those fluids? 2/3, they're inside of the cell, but then 1/3 is extracellular fluid. So now we're going to see what is inside of the cell and what is outside of the cell. Okay. So here is your cell. This is your nucleus. Fluid inside of the cell, you call that intracellular fluid. And then fluid outside of the cell, you call that extracellular fluid, but you have different types of extracellular fluids, right? So you have your interstitial fluid that is going between the cells. And then you have plasma that is running with your blood. So inside the fluids, what you're going to find inside the cells, you're going to find nutrients, electrolytes, and oxygen and sugars, okay? And waste products that are given from that cell, those are going to be like carbon dioxide and urea and some other byproducts. These are examples. Okay. So that brings us to the plasma membrane. So here's a presentation of the plasma membrane. And basically, what is it used for? Well, it's a very good barrier to keep the cell alive. And at the same time, it's a very dynamic thing because it has several functions. First of all, we have seen before that this is made of lipids. So here we have your phospholipids, right? So it's a phospholipid bilayer, so the 2 layers of those phospholipids. And among those lipids, you're going to find proteins that are embedded. Sometimes the protein will go all across the bilayer, so that is called integral protein. Sometimes the proteins are sitting only on one side, okay? So that is called peripheral protein. Okay, so integral protein or peripheral protein. And we see that the outside of the cell membrane, they also have those sugar components that you call glycocalyx, right? So those are made up of sugars that are helping the cell with all sorts of functions of communication and interaction outside of the cell. And when cells get together and get close to each other, they are going to interact by the cell junctions that we have here in the following slides. Right? So here is just an example, just putting in one slide the components of what you're going to find in the cell. And yes, there is a video that you can click and watch. Okay, so components of the plasma membrane. What you're going to have, you have a lot of proteins, you have phospholipids, you have cholesterol, and you're also going to have the carbohydrates, but those are really like to the outside of the cell. Okay. So you're in the structure of the plasma membrane. So we know we have your phospholipid. The phospholipid has a polar head that includes the phosphate group, and it has this non-polar body. Okay, so that's your hydrophobic tail. So phospholipids, when they get together, they do it without the need of any energy. They do it spontaneously, okay? So they get together and they form this bilayer just naturally. So the inside of the layer is the lipid part, and then the outsides, they are going to interact with water. So that's your hydrophilic part. Now, what kind of phospholipids do you have that are present in this membrane? You can have saturated phospholipids, and in that case, your carbon bonds are going to be all single bonds. Okay, so all single bonds for saturated lipids. And you can have also unsaturated lipids. So in that case, you're going to have at least one double bond among the carbon. No triple bonds in naturally occurring molecules in the body, okay, as a general rule. So if you have a double bond, that is your unsaturated fat. Okay. So 3 types of lipids in your membrane: phospholipid, cholesterol, and glycolipid. Oh, look at that, another point for you. Phospholipids orient themselves in aqueous solutions such that, oh, now what is happening here? You have polar heads, they are pointed where? Well, not to the inside. You have non-polar tails that are pointing to the inside. So let's pick up the correct answer here.
All right, time has started. Fifteen seconds. Five seconds. All right, and the answer?
All right,
D.
what is the answer here?
They said D.
D. Polar heads face the interior and the exterior of the cell with the tails forming the center of the membrane. Okay, that's fine. Your answer is D. Okay, and then we're going to look at tight junctions. So just 1 second. Now is 6:39. I need to take a 5 minutes break. So I'll give the class exactly 5 minutes to take a break and stretch your legs. And I'll be back here. I'll put it in the timer. If you want to, go to the restroom or take a sip of water, I'll give you 5 minutes starting now. Can you keep track of time there?
It is 5:40. You have until 5:45.
All right, 5 minutes break. Be back.
Yes. By the by, our first exam is in 2 weeks from today. No, not November. What is the month? September 10th. No, yes, and that's a good point. All of our exams are usually on Mondays, but this one is on Wednesday, according to the syllabus, because we have Labor Day the week before, so we're missing next month. Is it this Monday? Yeah, no Labor Day. Yes, Labor Day, no class. So we don't have lecture on Monday, this upcoming Monday, I believe, September, whatever, yes. The first Monday in September is always Labor Day, which means we don't have class. Yes. Yeah, you. Yes, you will need Scantrons, guys. The blue Scantron that fits like 100 bubbles. I know they give them for free at the student union. I think it's the same place where I get them as a graduate student, but you go up to the second, is it the second floor? I'm just going to tell you where I get mine. You go up to the second floor into like the student services, something. Yeah, in the student union, you go up to the second floor. They make you like scan some QR code, and then they fill out the form, and you can take 2 Scantrons and 2 pencils. Speaking of, you will need a pencil. If you want to risk it and use a mechanical pencil, do it. I think it'll be fine. But definitely a pencil and not a pen. And I'm going to put out a little form about how to fill in your Scantron, because if you fill it out wrong, I will not know who you are. Because Scantron doesn't put these into Canvas for us. It just gives us an Excel sheet. And so if you put your name in wrong, I'm not going to know who you are. So I will upload that, and I hope everybody looks at it so that you know how to fill out your form. Did you have a question? 50. In person, which means you have to show up to class. All of you, including the people who are not here. Hold on. Yeah, go. Yeah, multiple choice. The syllabus will tell you that. But it will be anything up to the point of the lecture. So like anything through that Monday. Yeah. It's in here unless you have SAS accommodations. But yes, come to this room at the scheduled class time, 5 p.m. I would show up before. And yeah, don't cheat. That's pretty a no-brainer there, but. And the class is only like, we're probably going to space you out so that nobody can look at each other's forms, but looking at other people's exams is cheating. Yes.
Oh, cheating. What was the question? What is considered cheating?
Oh, yeah. Dr. Rosari, just to confirm, the first exam is on Wednesday the 10th, correct?
Yes, I think I had a wrong date on the Canvas, so it's going to be on Wednesday.
So the syllabus is correct. Her announcement is currently incorrect, yes.
Yeah, he's on Wednesday. I usually give it on Mondays, but this time it's going to be on Wednesday. Okay, so coming back here to the cells, hopefully everybody's back in the class. You still have more extra points. Okay, so here, coming back here to the cells, when the cells get together, they form this interaction among each other, okay? So you have proteins on the surface of those cells, and those proteins fuse the cells to each other, kind of like stitching them together, okay? So this stitching of the cells make this type of impermeable junction that you find right there. So those are called tight junctions. They're really, really tight, fusing the cells together. So this is very important for blocking the movement of cells or movement of things among the cells, between the cells. Okay, so most things will not be able to pass through those tight junctions. Okay, so this would be very important in tissues, for example, like your skin, because you don't want things to be coming out of your body and going through the tight junctions. Then other forms. Here, come on, today. Okay. Other forms of junctions that you have, those are the gap junctions. Right? So those are like little straws that you put between the cells. And they're important for communication among the cells. Now, the proteins that make up the gap junctions, those are called connexons. Okay, so those are like little straws that make the connection. And for some reason, my videos are not working in this PowerPoint. Probably there was some kind of update and messed up on my videos, my little, illustrations here. But you can imagine these gap junctions, they make a gap between the cells, between one cell and the next cell, okay? And they allow movement of things through those little straws. And what is it can go through? You have ions that will diffuse across, and you also have electrical signals that can go across those gap junctions. And this is a very important feature in cells, for example, of your heart. Okay, so gap junctions are actually going to allow the electrical communication of your heart cells so that all your parts of your heart can beat together. Okay? In syncytium. Okay. So what is it that's going to go through the membrane? You know the membrane is not going to let just anything go through, otherwise there'll be no use for having a membrane in the first place, right? So it's going to select things. When you need energy to transport things through the membrane, you call that active transport. And what is the energy that you're going to use? Well, it's ATP. When you don't need energy, you're going to call that passive transport. Okay. So now let's see here types of transports through the membrane. You're going to have passive or active, okay? Active transport is going to require energy. And passive transport, here are examples of passive transport, that's not going to need energy. So we're going to have diffusion or facilitated diffusion or osmosis. Then vesicular transport is really going to use the membrane itself with fusion vesicles or budding vesicles. They're going to interact with the cell membrane. So here we're going to start looking at what is this type of transport. And yeah, this was another video that, of course, is not going to work. It's not working. Okay, so passive transport, no energy input. What kind of passive transports you can have? You can have simple diffusion or facilitated diffusion. In simple diffusion, you don't need a protein for that. The thing can just diffuse on its own. In facilitated diffusion, you need a channel. So that channel is either going to be closed or open. When that channel is open, you're going to allow diffusion to go from higher concentration of stuff to lower concentration of stuff, okay? And on the other hand, you can have osmosis. Osmosis has to do only with the movement of water. So in that case, water is going to go from an area of high concentration to area of low concentration. We're talking about water. So if you look here at this solution over here, if you look at the bottom part of here, you have more water here than you have over there. So the water is going to move that way. Okay, so that's osmosis. And of course, that would help if you have a semi-permeable membrane in the middle over here. That's my semi-permeable membrane. So water will go from high concentration of water to low concentration of water, and that is osmosis. Now, if you're talking about diffusion, you're going to look at those particles. Particles everywhere over here, the green stuff here. You see the top part has a lot more particles than your bottom part. So diffusion of the particles would be from areas of high concentration of stuff, that is not water, to areas of low concentration of that stuff. So that's diffusion. Now, example of a facilitated diffusion is when you put a channel over here, okay? So if those particles use a channel to go through, that is called facilitated diffusion. Okay. So here, what is going to influence the movement of molecules? What is it going to influence this diffusion stuff? So first thing we're going to look at here is the concentration of things that you have, all right? So when you have things that are highly concentrated, it will diffuse faster to the area that is not concentrated at all. So here you see this example of high concentration going to low concentration. Also, the size of the thing that is diffusing. If you have this bulky molecule, it's not going to move very easily. It's going to take its time. If you have something tiny, that will really wiggle very quickly and get to the area that is where it's less concentrated. Okay, so molecular size is also important. The distance that the thing has to travel also is taken into consideration. Okay, so if you're looking at how things are diffusing from here all the way to the other side of the beaker, that the first part of the beaker, of course, is going to have more concentration and more diffusion than at the other side. So distance and area, those are correlated. Oh, this is a good one. The temperature that this thing is held on. So yeah, if you look at extremes, for example, if this water is not really liquid and is becoming crystallized, like almost frozen, then most likely you're not going to have a lot of diffusion. Okay, so things are not going to diffuse very easily if your molecules are not moving around a lot. When you get this water warm, like at body temperature, then you have better diffusion of solutes throughout the aqueous environment. Another thing that's very important is this lipid solubility, because lipids do not really dissolve in water very easily. So if you have a high content of lipid in your molecules, then most likely you're not going to have a lot of diffusion, because the lipids are going to tend to aggregate in one place, and they tend to just get together instead of just diffuse all around. Okay. Okay, let me see. What are the things that are going to go through membranes? All right, this is very important too, because some molecules can just go through the membrane and not worry about anything. So the molecules that can just pass through the membrane, those are going to be small lipid-soluble molecules. And so we're talking about the membrane, going through the membrane, not just diffusion in water, okay? When you're going to go through the membrane, you're going to have tiny lipid-like molecules that are nonpolar. Those can go through the cell membrane without any problem. So a couple of things can go through the membrane. Very small nonpolar molecules and very small lipid-soluble molecules. I have more on that in a few slides. Okay. If you have bigger non-lipid molecules, those are not really going to go through without an issue. They're going to need to be carried. They're going to need a carrier protein, okay? So if they need a channel, like what we see here, if they need a channel, then that is called facilitated diffusion. Okay, you got to remember that, okay? Now, remember, osmosis is the movement of water. Okay, so water molecules will go from the area where you have more water to the area that has less water. So if you look at the cytoplasm and the inside of the cell, you're going to have less water than you have at the outside, at the interstitial fluid. Okay, so water molecules, since you have more water outside of the cell, those are going to go through the membrane via special channels. They're called aquaporins. Okay, so pores of water, aquaporins. So water will go through aquaporins, and they're going to go to the inside of the cell through diffusion, right? So here is the name of the channel that allow water transportation. So those are your aquaporin channels. All right. So with the simple diffusion here, we see that certain things will just go through the membrane without the need of a channel, right? So these are very important things that you have to remember. What can go through the membrane? Look at this. You gotta know that, okay? First thing is gas. Gases will go through the membrane. What gases? Oxygen and carbon dioxide. Thank goodness, those will really pass through the membrane. So oxygen shoots through the membrane. Carbon dioxide will shoot through the membrane, okay? From areas of higher concentration to areas of lower concentration. That will make sense when you start looking at respiration, which is actually next semester, right? Next term. When you start looking at respiration, you know your body needs oxygen. So you're going to allow oxygen to go from higher concentration in your lungs to the lower concentration of oxygen that is in solution in your blood. So oxygen can freely go through the membrane and get to your cells very quickly. Okay. On the other hand, when you look at the tissues, you're going to have higher concentration of carbon dioxide in your tissues just because of regular metabolism of cellular respiration. So you need to get rid of the carbon dioxide. So your tissues will allow that gas to go from inside of the cell to the outside of the cell and reach the blood and be carried away from your tissues and get into the lungs. When that gets to your lungs, then you're going to have less carbon dioxide outside of your body, right, in the air, than you have in your tissues, so that you're going to be freely diffusing the carbon dioxide. So absolutely remember what is the one thing that is going to go through the membrane without any restriction? That is the gas. So gases will do that. Another type of molecule that will go through the membrane because they are very small and they are lipid soluble, that's going to be steroid hormones, right? So hormones that are deriving from cholesterol. So those are your steroid hormones. Those can really diffuse through the membrane and find their receptor in the cytoplasm of the target cells. Okay, target cells will have the receptors for those. Okay, so here is your example of very small lipid-soluble molecules that will just go through that membrane and get to the cytoplasm. Okay, so here is just another depiction of what happens in diffusion. Okay, so you're going to get a lot of diffusion when you have a higher concentration gradient among those two areas. Now, you also have molecular diffusion going from this side to the other side because molecules are bouncing around all the time and they move everywhere. But look at this, the bulk movement is going to be from the higher concentration to the lower concentration of solutes. And very little number of molecules is going to go back to the other side, but it's really overpowered by the whole bunch of molecules is going this way. So here, when you take away the ones that are going backwards, you get the net diffusion going in direction of higher concentration to lower concentration. Okay. When both of the sides have the same concentration, then you don't have diffusion anymore because the same number of molecules that are going to go this way is the same number of molecules that are going to go to the other way. So these two movements just cancel out. Okay, so here's a cool picture showing diffusion of something that has color in the beaker. And here is another question for you. I told you, tons of questions today. So lucky those of you who actually stayed in the class. All right, question. Which of the following is not an example of simple diffusion? Option A is gas exchange in your lungs. B, a dissolving sugar cube. C, popcorn odor permeating the room. Oh, I'm getting hungry. D, a white blood cell engulfing a bacterium.
All right, time is starting now. Twenty seconds. Ten seconds. Most people were able to answer, so maybe restart your iClicker device. All right, what's the answer, everybody? Everybody says D.
Yeah, that is, what do you call this process? Phagocytosis. Okay. So now let's look at facilitated diffusion. Again, this thing will go from area of high concentration to area of low concentration, but now this thing is too bulky. It's not going to diffuse through the membrane, okay? So it needs a protein. It needs a channel for that. So the channel can work in a certain way, like changing conformation when the solute or when this substrate is present, okay, in order to deliver that solute to the other side of the cell, okay. Or the channel can just be open at certain times and allow this thing to diffuse across just because the channel opens. And of course, these channels have to be regulated because usually you just don't want diffusion of stuff in or out of the cell all the time. So you need to regulate the opening of those channels, okay? Well, there are some channels that are open all the time. Like there are very few of those. Those are called leakage channels, right? And those are important for, in one case, for proper functioning of your heart cells, of cardiac cells, right? So you always have some leakage channels that are going to allow certain current to go through, and they're going to allow your heart cells to beat by themselves on their own without an outside nervous input. Okay, so these channels are always on, leakage channels. And then you have some other channels that are gated channels. So those are the ones that are controlled all the time. So gated channels, they need to be controlled either by certain chemicals that can bind to the outside of those channels, or they can be controlled by electrical signals, that they will feel the change in the electrical signal, either inside or outside of the membrane, usually inside of the membrane, and the shock will make the channel open or close, okay? So leakage channels are always open, very few of them. Gated channels are controlled to open at a certain time because of the presence of either chemicals or electrical signal. And yes, you know, if you know me and you have taken other classes with me or you heard about how I do things in the class, you know that if something is red and bold or underlined, that's the kind of stuff that I like to use, like to ask in exams. Okay. So here's another slide showing the idea of osmosis, all right? Remember, osmosis is movement of water. So you always have to look at where is it that you have more water and how the water is going to move around, okay? So osmosis is a movement of water from high concentration to low concentration. And that brings us to this idea of tonicity, okay? So what is happening over here? If you look at your plasma, you're going to see that it has the same concentration of solutes as the cytoplasm of the cells that are floating around in the plasma, okay? So you have certain proteins in the plasma that will make it isotonic solution, right? So here's your first term. Isotonic solution, it has the same concentration inside the cell and outside of the cell where the cell is found. So of course, tonicity has to do with cells, right? So isotonic solution, the inside of the cell has the same concentration of stuff. Another name for that is osmolarity, okay? Same concentration of stuff, both inside and outside of the cell. So the total volume of your cell is not changed. So here is your example of a red blood cell in an isotonic solution. So water going in is the same amount of water that is going out, okay? So there is no, well, there is osmosis going in and out, but it's the same amount of water going in and the same amount of water going out. So the cell stays in the same shape. Now, when you look at a hypertonic solution, hypertonic, that means you have more stuff outside of the cells than you have in the inside of the cells, okay? So that's a problem because when you have a lot of stuff outside of the cells, that stuff is going to pull the water from the inside of the cell. So you're going to have a net movement, net osmotic movement from the inside of the cell to the outside. So your cell is going to shrink. Okay, your cell is going to let the water out, and the cell is going to, to have this weird shape in case of this red blood cell over here. It's going to shrink. And then the opposite happens when you have a hypotonic solution. Okay, so here you look at a hypotonic solution in which you have less solute outside of the cell, and you have more solutes inside of the cell. So what's going to happen, the water is going to go from the outside, there is more water, to the inside of the cell, and your cell is going to blow up. Okay? So cells will blow up if you dump them in hypotonic solution. Okay? Let's just highlight here with something you can actually read through. Hypotonic solution. And look at that, another question. Oh, again, I just gave you the answer. Red blood cell placed in a container of distilled water. So what kind of water is that? Does it have anything in that water if it's just distilled water, clean water? That is your hypotonic solution. So the cell will what water via what kind of mechanism? All right, let's start that. Let's start that eye clicker.
All right, time has started.
We can do the timer thing here. That's essential. Is the timer backwards?
15 seconds. Just put something in, guys. You get half credit just for putting something in. 3, 2,
Ooh, I'm
1.
on.
All right, correct answer. They said B.
Okay, so hypotonic solution, it will gain water by osmosis. Yeah, you're right. Oh, great. Now my dog is going crazy here. Go, go, go out. This home business is teaching from it is not what I like to do. Oh, by the way, next week I'm gonna be in the US. Yay! But I will not be on time to teach this class because I'm getting there on Friday morning. But yes, so next week is the last two lectures that we are gonna have via Zoom, and then I'm gonna be back home.
Dr. Waziri, just a reminder that this upcoming Monday is Labor Day, so there's no class.
Oh, yay! Okay. So we're gonna have class on Wednesday, and then and then for the following week, we're gonna be there. Well, it's not Labor Day here, so I forgot about Labor Day. Okay, so one more week of this torture, and then I'll be there. All right, so here again, when you go through facilitated diffusion, right? Still following the rules of diffusion, right? So we're gonna go from high concentration to low concentration, but you need a protein for that. Okay, you need a channel for that. Okay, what is gonna affect this rate of transport? You are going to have the protein. So the number of proteins that are embedded on that membrane, those are going to interfere with the rate of diffusion. If you have only one protein over there as a channel, you're going to have one molecule going at a time. If you put 100 of those, then 100 molecules can go at a time. So you're going to have faster diffusion. Okay. Another thing that is going to influence how much stuff is on the higher concentration. So the amount of the substance in the higher concentration, the more gradient that you have, the faster the diffusion that you're going to have. All right, and then we get to the last form of transport here, which is your active transport. And you still have other subcategories of active transport. You can have primary active or secondary active. So let's look here first at your primary active transport. You're going to have two types. You can have either one second. Yeah, of course, I got to be interrupted here. So two major types of membrane transport, okay? you're going to have active transport. Also, vesicular transport is also considered active, but is more in the secondary active. Both of them are going to need ATP. So if you need energy, that is considered active transport. So what is going to happen here? When you have substances that are too big to go through channels, you need to push them through this transport mechanism. Yeah, I have to have my helper here to finish the class. She can't take it anymore. All right. So when you have protein solutes that are too large, you're going to have to use ATP to push them through. Okay. When you have something that is very bulky and that is not a lipid, you're also going to need energy to push them through. Okay. Another thing that is very important, that is the case here with your sodium potassium pump, is when you're pushing stuff against their concentration gradient. Make a note of that, because that is very, very important. When you're moving stuff against their concentration gradient. Okay. So what is happening over here? You have this active transport that is pushing some ions to the outside of the cell and other ions to the inside of the cell. Now let's look what is happening. You have here sodium ions. You have sodium ions. You look inside of the cell in the cytoplasm. And overall, let's count here. 1, 2, 3, 4, 5, 6. You look at outside of the cell, at the concentration of interstitial fluid, and look at this, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10 to 6. So which side is more concentrated for sodium? Is it inside of the cell or is it interstitial fluid? Anybody?
Interstitial fluid, people
Yeah,
are saying.
outside of the cell, you have more sodium particles. So this channel here is going to push more sodium particles to the outside of the cell. You say, oh, but I already have enough. It doesn't matter. This guy is going to use energy, is going to force sodium to the outside of the cell against the concentration gradient. And then you look at potassium. You have more potassium inside of the cell than you have outside. And this guy here is going to push potassium to the inside of the cell against the concentration gradient. Okay? So this is pushing sodium out, is pushing potassium in. How much of those? Is going to push 3 sodium out and is going to push 2 potassium in. Okay? 2 potassium in and 3 sodiums out. Okay, that's your sodium potassium pump. Again, the movie is not working. All right. So you got to know about that. You got to know about everything about the sodium potassium pump. Okay. They're working against the concentration gradient. They're using energy. Like what kind of energy? They're using ATP. Okay. And you're going to have ATP hydrolysis, right? So what's going to happen here? You look at this channel. And this channel is pushing something out, another thing in. So this is called an antiporter. So this is an antiporter channel. Push one thing in, another thing out. Okay? So let's see what's going to happen here. The conformation of the channel is going to change. Okay? When the channel is open to the inside of the cell, it is not using ATP energy. So it's naturally open to the inside of the cell. When that happens, it has this conformation that is binding 3 sodiums coming from the inside of the cell. Okay, so it binds there. Now you use ATP energy, you put an ATP on that side of this channel and you change the conformation. That change in conformation is going to push the sodium to the outside of the cell. It's going to dump it out there. And is going to change the conformation of the binding site. Now you are able to bind 2 potassiums. And the potassium is going to be bound there while the phosphate group is bound to the membrane, is bound to the channel. This potassium binds there and nothing is happening. When you push away your phosphate from the inside of this channel, then the conformation is going to change again. Because remember, no phosphate means this thing is open. It's such a pain in the neck. So the thing being open you're going to change the conformation again, and you're going to dump your potassium to the inside of the cell. So everybody going to know how sodium potassium pump works, okay? No phosphate here is opening to the inside. Bind sodium, add the phosphate, dump the sodium outside. At the same time, you're picking up potassium. Kick out the phosphate, you dump the potassium inside of the cell. How many sodiums go out? Do I hear the answer?
3.
Yes. How many potassiums go in? All right. So make sure you know that. Okay. So we have 2 minutes here. I'm going to stop at this slide before we get to the electrochemical gradient, because this is another concept that it takes a bit also time to go through, but it has to do with the sodium potassium pump. Okay, we're going to pick up from here on the next lecture, and I'm going to hang around over here with my little pain in the neck doggy and wait if you guys want to come to the podium and ask questions or make comments.
Oh my God, but I was right. But you changed your answer. Because you
What?
took peer pressure? Jeez. Hold your own, girl.
What's going on? Peer pressure, lost points, something? Did I hear it right?
I am peer pressuring you.
You see, now that I'm not talking anymore, then she's quiet. You're pain in the neck, you know that?
Those shows as if I'm like I have like I think one absence. I think that was from like the first day that we did, and there was some issues.
Yeah, she hasn't put them onto canvas yet, but when she does, it'll rain.
Yeah.
When she puts them on, she's gonna not count anything from the day one,
Okay.
and then she'll fix the thing that I did wrong on the second day. So everyone will get the point.
Because I think like besides the first day when there were issues, like everything else was fine.
Okay, good. And are you the one, like, you're one with the remote? Or is that not,
I
yeah?
have a
Okay,
remote.
and it's been working, yeah? I don't know what happened the first class, but
Yeah, I don't. It was just saying no base, which was confusing.
I had it plugged in there,
I
I
had
plugged it in
to
there.
use the remote. My bio principal last semester, and it was working fine.
Yeah.
So I was confused.
Oh, I figured out what happened with the first day with the no base. What
there was,
happened?
I had the base. Well, not like the first day of class, but the first day that we tried the iClicker, I had the
Yeah.
base.
Yeah, but when it wasn't working, the
Yeah,
site,
I had it plugged in, it just wasn't doing anything, so I'm not sure. But
yeah,
it all
the
works now,
site,
so
you see, the site, what happened was that the site itself had changed the configuration of the base to BB instead of AA. So when I went in and I was checking what could possibly go wrong, that I found that. Then I changed to AA, so that's why it's working again.
okay.
Yeah.
But like I said, I also stopped plugging things into the class computer because
Doesn't work.
yeah, I'm just not gonna do that.
Mhm.
Yes. All right, good to go, Doc.
Yeah, I guess we are. Anybody else has a question or something or
Nope, everybody's out.
comment? Everybody's out? All right, so I'll see you. When do I see you? Next week, Wednesday.
Wednesday, yes.
So one more Zoom lecture and we're done with this. Yay.
Well, we've got it down now, but yes, I will be excited to be done.
Yeah, especially as you've seen, I have a lot of interruptions here.
Yeah,
And
but people enjoyed seeing your dog. It actually helped them pay attention. So.
yeah, then she'll be holding the class attention. I'll bring her to class sometime.
I'm sure people would love that.
Yeah, she's
Yay.
a very good little service dog. Yeah, look at that face. I mean, how can you not love that face with the big ears? All right, bye. See you.
Okay, see ya.
Thank you. Bye.